Hello everybody. We're looking at chapter 4 part 2, and we're going to take a look at a little more in reference to the bacteria that we've been talking about in chapter 4 here. We talked about a variety of things in chapter 4 part 1, and in chapter 4 part 2 I'm going to expand on some of those and talk about some of the concepts related to a handful of these structures and features of the bacteria we mentioned in part 1. So, for example, I'm going to talk about some of the genetic transfer that happens. We're going to talk about how biofilms are formed. We're going to talk about capsules and their medical significance. And in the last part, we're actually going to introduce something different called, about bacterial endospores. And that's a different group of bacteria that have a unique, really hardy survival feature. And that is something that we'll kind of introduce for the first time here. So we're going to build on some of the last topics and then add a little bit new here at the end. So in part 4, part 1, chapter 4, part 1, we started talking about some of the genetic material. Pull that up real quick here. Didn't mention much of it, but I mentioned that a lot of the genetic material in a cell, we're looking at figure 4.1 here, a lot of the genetic material in a cell is found in what's known as the bacterial chromosome. And if you look here, this is pointing to all of this here in the middle of the cell, which is the bulk of the genetic material. So the DNA is what we're talking about, organized in what's called a chromosome. And this is what makes up the majority of the genetic material. Now, there's another little piece of genetic material that occurs in a handful, or not a handful, but actually quite a wide variety of bacteria. It's not a consistent, it's kind of a hit and miss, and there's different variations of these. But what's often found, I should say, are these smaller pieces of DNA called a bacterial plasmid. If you look, we didn't really talk about this last time, but all the way down here is pointing to a plasmid. Really what that is just an extra piece of DNA that's got an extra small number of genes compared to the chromosome. It can be a dozen or more. Usually it's just a couple, but it's a small number in comparison to the several thousand found here in the chromosome. So these genes can come in a wide variety of forms. There are lots of different types of plasmids, and you almost think of them like a little CD or like a little disc of some kind, and they have a relatively small amount of information. It's not some mega hard drive, right? It's a relatively small amount of information, and it can be transferred from one area to another. So it's like a little disk. It can hold a small amount of information and then be transferred around, okay? Like a CD. But in this case, they hold genetic material, and that is called the bacterial plasmid. So I mentioned that, although very briefly, in the last section. So here I want to expand upon that. So what they are, as I said, a small piece of genetic material, and they're referred to as non-essential. And let me just clarify that by saying that the bacterial chromosome, that larger piece I mentioned that's going to contain several thousand genes, it is considered essential. Essential genetic material. The plasma, and really what that means, let me just clarify, what that means is it's necessary for basic life functions. Think about it that way. In other words, it can't really survive if that chromosome becomes damaged or mutated to a certain extent. The plasmid, on the other hand, is considered non-essential, and that's because it is unnecessary. It's really the opposite here of unnecessary for basic life functions. Oopsie. But can be beneficial under certain circumstances. Under certain circumstances. Okay. So that's kind of the difference. The chromosome is the main kind of core center of information in the cell, whereas a plasmid is like this little extra piece of information. And what's key about that is that it can be transferred from one bacteria to another. So it's like an extra little set of information, that can actually be swapped from one cell to another. And when that plasmid is swapped, the genetic information goes right along with it. So when bacteria are transferring these plasmids, what they're actually really doing is they're transferring characteristics from one cell to another in real time. This is something that doesn't happen in this type of manner in different areas of life. There are other places, but this is somewhat unique to the microscopic realm of our world. And to give you kind of an idea of an analogy of how odd this really is, imagine two people standing next to each other, holding hands, and being able to pass different attributes back and forth to each other. You know, maybe like hair features or different body features like muscle mass or fat tissue, or even being able to pass different abilities back and forth, like learning a different language or being able to play a musical instrument, something like that. Very, science fiction-y, right? That's the idea. In our world, the macroscopic world, that is completely just fantasy. Might make for a good movie, but at the microbial world, this is something that actually happens in reality. Now, it's just not something quite as amazing as what we might do on our level, but on their level, it's that same kind of thing. What we're talking about being transferred, for example, are things like antibiotic resistance, the ability to resist a drug that might otherwise kill you. That's actually a physical trait that bacteria can acquire through the transfer of something like a plasmid. There are other ways that it's done, but that's one of the primary mechanisms in which that can take place. So when we're transferring traits or transferring genetic information, that is, we're actually transferring traits. And the example I'll use is antibiotic resistance, because that's going to be the most significant one that goes along with that sort of genetic transfer. So the plasmid is responsible for the genetic materials. It carries the genetic material, such as an antibiotic resistant gene, and it can be transferred. The process of transferring it is called conjugation, and that's something we talked about back in chapter 4 as well. Going back to chapter 4 here, chapter 4, part 1. I talked about the pilus or the pili, the attachment structure between two or more bacteria, and I talked about how this is used in the process of conjugation. So what conjugation really is it's a process by which bacteria link using the pili and then exchange plasmids back and forth and swap that genetic material. Now, the idea behind this is that, as I've said already, any trait associated with that plasmid will now be transferred along with that process of conjugation. And when we talk about how this works, this is pretty unique in that it's done in real time. And what I mean by that is that we're talking about a process called horizontal gene transmission. And I just want to compare that. So here, talk more about that here. Horizontal gene transmission is the process of transferring a gene from one mature cell to another mature cell, often sometimes referred to as a parent cell. We can contrast that with what's called vertical transmission. So let me do that real quick. Pull up a figure here. So when a cell is passing along information to another cell, the idea behind that is some sort of genetic transfer. Okay, so we're talking about a genetic transfer when we'll say cell A here passes along information to another cell. We'll call this one cell B. Now, when that takes place in a vertical transmission standpoint, what's really happening is cell A is replicating and is actually going to make a copy of itself, and cell A is going to transform into a copy of itself, which is now going to become cell B, and we're going to have two copies of cell B. So essentially cell A is transforming into cell B. Now, any of the DNA associated with cell A is going to be copied in the process of DNA replication and through the process of mitosis. And unless we're talking about sex cells, which would be meiosis, Then through mitosis, we're talking about the DNA being split evenly into each of these two cells here, what we're calling B. Now, technically, the technical terminology here, rather than A and B, is that this is actually called a parent cell, and these are referred to down here as daughter cells. And this process here is vertical transmission. I'll let you write in the word vertical here. But so when DNA information is passed on from a parent cell to a daughter cell through cell division and making a copy of the cell, that is referred to as vertical transmission. Now, if cell B turns out to be a different cell, such as another adult cell, then we refer to that as horizontal. And so here what's happening is this cell A isn't replicating. It's not duplicating itself in kind of the natural process of passing on information to another cell through cell division and mitosis or meiosis. It's not doing any of that. It's either usually it's just some of the DNA is being transferred directly from one parent cell really to another parent cell. Sometimes you might just refer to it as an adult cell or a mature cell or something sometimes they refer to it as a recipient cell. A lot of times this is actually referred to as a donor and a recipient. Let me pull up a figure here. Here's a figure showing a donor and a recipient. The idea is that you have one cell that contains a plasmid or a piece of DNA, and another cell that doesn't, and in the process of conjugation, or in this case we're talking about horizontal gene transfer, the DNA is passed from that cell, from one cell to another, the donor to the recipient. So here we can call this cell a donor, and here we can call this cell a recipient. Okay. So, in general, and just to clarify here, in general, when a donor cell passes DNA to a recipient, in general, that process is referred to as horizontal gene transfer. So there's a variety of ways this takes place, and we're not going to talk about all those right now, but I'm talking about horizontal gene transfer so that then we can talk about the one example that we do want to mention, which is conjugation. Okay, so conjugation, going back here, is the process by which the plasmid of a bacteria is specifically shared with another bacteria in the process of linking up their pili together. Okay, so that's an example of horizontal gene transfer that takes place between bacteria. All right, and that's kind of the idea behind talking about horizontal gene transmission. All right, so I use the vertical transmission to contrast the way in which DNA is usually passed along. It's usually passed along from one parent cell to the daughter cells during cell division and cell replication. So when we talk about the plasmid, then again, let me go back to my drawing here just real quick, or actually let's pull up this figure just to clarify. We're talking about this plasmid here being transferred. So this picture here shows the donor, here's the pilus, they link up, and then now that plasmid down here, they're showing the plasmid being replicated, making a duplicate, and now the duplicate here being shown here is passed into the recipient cell, and after the process is complete, they each now have a full plasmid, whereas initially they only had one. And why this is so significant is because what can happen through this process is a gene of interest, or a gene that is significant to the survival of a population, can be easily transferred amongst a wide variety of bacteria relatively quickly. And this doesn't always have to take place between the same species. This figure here, for example, is showing an antibiotic resistant cell passing along resistance to what would be called a sensitive cell, meaning that it would normally be killed by the antibiotic. So it's sensitive to the effect of the antibiotic, meaning it kills it. So in the idea of where plasmids are significant to health is that you're getting cells with resistance passing along the characteristic to cells without it. And what can happen is we get an increased amplification of that gene in the population. So Here, here's a figure showing a similar process in which you have the plasmid initially being transferred to a cell that was the recipient. And then that recipient now goes on to duplicate itself and make more copies through vertical. So we have really both ways of amplification. Normally, without the plasmid, you have just vertical gene transfer, and you have the normal process of cell division, in which one cell can theoretically make millions, if not billions, of copies of itself. So you already have a huge number of cells dividing through a kind of a natural process of one cell making two cells, and so on. Now you're introducing this horizontal, in which now a whole new lineage of cells will pick up that trait, whereas before only certain lineages would have it. So in theory, if you could wipe out the lineage of cells that had the resistance, then you could theoretically see an end to antibiotic resistance. The problem is, at least, and there are really many problems to be fair, but one problem is that these genes keep getting transferred from one lineage to another, and now you have this horizontal spread as well as the vertical spread through replication, and it's a real problem because the resistance mechanisms are just too difficult to get rid of. There's too many of them, and they're too easily transferred, and you take the replication ability, how fast they can replicate and vertically transfer those genes anyway, the process really becomes exponentially worse than it would be if you had just one mechanism or the other. And so that's what this slide here is kind of talking about, is how the significance of plasmids really relates to antibiotic resistance. And that's really the primary example to make here. Other examples really get into the more technical aspects of microbiology that are a little harder to associate in a more straightforward way. So that's really the example of how plasmids are significant, and how that process of conjugation, the spread of the plasmid, is significant in the medical context. Now, there's another context to which plasmids are pretty significant. Plasmids are also significant to biotechnology. The reason a plasmid is significant to biotechnology is because scientists can actually add new genes to a plasmid. It can actually modify a plasmid and add a new gene of interest into it. Let me pull up a figure to show you. So here's a figure showing a bacterial plasmid, and what started happening early on in the 1950s to the 1970s was they were studying these bacterial plasmids, and initially they just found out that they were there. Then they eventually figured out how to extract them, and then they started figuring out how to actually cut them and modify the plasmid. So using these molecular tools, they could actually cut open the plasmid and then literally add a new gene directly into the plasmid. So now what you're doing is adding more genetic material than what was initially there. So then you can actually take the plasmid and put it into a new bacteria, and you can actually then get that bacteria to acquire a trait that you added in reference to the genetic material. So there's a number of examples here, and if you want to look at this website here, I let you look at the link up here if you want to copy that. This website here, it shows a number of different examples here of how this type of technology is used, but this is the example I want to focus on. What they did in this particular example is they found the gene for human insulin. And it turns out that insulin, and you may be familiar with insulin and how it works, and a lot of you may work in hospitals where you give insulin to patients. The idea is insulin helps us regulate blood sugar. And what it is actually a protein. And the protein is produced by information in your DNA. So there's literally a gene in the DNA that basically tells your cells how to make insulin, and using that genetic information, the body produces its own insulin. For a variety of reasons, people either don't produce enough insulin or they don't produce any at all, and they need to take insulin supplementation in order to help regulate their blood sugar. So initially, insulin was derived from slaughterhouses when people needed extra, but that was deemed to be not the best way to do it. So what they ultimately came up with, and this is somewhat controversial, they ultimately came up with a way as I mentioned, to extract a gene from the human chromosomes and then put that gene into a bacterial plasmid. And then what they did was put the plasmid into a bacteria, an E. coli bacteria, and then literally what ends up happening is the E. coli actually makes human insulin. It actually converts the human gene into a protein inside the bacteria. And the way this works is it turns out that the bacteria doesn't care where the gene comes from. It doesn't care if the gene comes initially from the bacteria or from a different organism. As long as you put the correct genetic information in there, it'll make the same protein whether it's in the human cell or the bacterial cell. So by taking the gene out of the human body, putting it into the plasmid, and then transferring that into the bacteria, you actually get the bacteria to make insulin for anybody who gets this process to work. Anybody who conducts this process done correctly can actually get bacteria to synthesize insulin. And then what they do is they do this on a large scale and extract the insulin and collect it after it's been purified. So there's a lot of details I'm leaving out here, of course, but that's the idea. So biotechnology, the ability to create new biological products like proteins and things like that, was really founded and established along the lines of genetic engineering. And the genetic engineering itself really started with the use of plasmids, and other discoveries that we'll talk about these later called restriction enzymes are pretty key, and a couple other things that were key to this whole process. But plasmids were really kind of the fundamental tool, the platform for which this process began. And so that's another reason why plasmids are significant. It really just goes back to this ability to swap traits between adult cells. It's really kind of almost like science fiction, really. Again, it is to you and I. But on a microbial level, it's reality. And I think it's important to emphasize how really odd that is. And it's a lot of people hear that and they just think, okay, whatever. And I always think to myself, like, I find that to be really mind-blowing, that bacteria have the ability to swap traits back and forth like that. And I think it's important to emphasize that really is a crazy kind of unusual thing that we just don't see on our level. And so it helps us understand why they're so hard to kill. And that's really what it comes down to. The antibiotics aren't that great anymore because they're actually adapting back towards us. And we think of them as these simple little mindless creatures, and they are. But little things like that give them a huge edge in being able to adapt to what we do in terms of drugs and things like that. So that's, I think, important to emphasize that really is a very unique and odd kind of thing that happens there. Now, the next part I want to talk about are biofilms. Biofilms are a way microbes can actually grow as a community and create a kind of a new little microenvironment around themselves. So a biofilm is a miniature secreted layer around a group of microbes that literally encloses them and creates this kind of micro environment around these individual communities of microbes. It turns out that this is a fairly common way that microbes live in the natural environment. It's not always obvious, but one of the things scientists have been learning, microbiologists have been learning, is that microbes in nature will tend to form some kind of relationship, such as the formation of a biofilm. And to clarify, this is an example of a symbiotic relationship between multiple microorganisms. We talked about symbiotic relationships in Chapter 1, Part 2. And this is important to recognize that this is an example of that amongst microorganisms. So really a biofilm is defined as a complex association that arises from a mixture of microorganisms growing together on a surface of a habitat. So the key words of that are complex association, mixture of microorganisms, and on a surface really is another key part. So what you have is this kind of secreted layer that comes out of a mix of microbes. That's the complex association. It's a secretion. I know it doesn't make any sense. It's a complex association of biochemical molecules is what that means. So it's this secretion of this complex biochemical mixture of molecules. It comes from a group of microbes, a mix of different types of microbes, and they do this on the surface of something. They don't do it within, they do it on top of a surface. So that's the other key part of that here. And another key part to connect back to chapter 4, part 1, is that the complex association, that mix of chemicals, really begins in most cases from the secretion of the glycocalyx layer, oftentimes the slime layer. So if we go back here just real quick to part 1, we talked about the glycocalyx, and we talked about the slime layer in the capsule. So the slime layer is a big part of the early foundation of the biofilm. So let's take a look at a few pictures here. Here's a biofilm. This is obviously an animation. But the idea is that what you have is a group of cells that start out on a surface, and what they begin to do is secrete a substance around themselves that will help stick them to the surface and kind of permanently attach them there, and also kind of start to form the basis of that film, that biofilm that they're forming. Now, what ends up happening, and this is a progression here over time, these can take weeks to form, it's not an automatic thing, but over time, more microbes will eventually come in to the film as they stick to it from the outside world, and this will contribute to a greater and greater enhancement of the biofilm and the formation of these structures here. So as that progresses and matures, we see that it starts to grow to an elevated height, and eventually what will happen is the cells within it will mature, and the film will actually rupture, and some of these cells will actually move on and actually migrate out of the film and go on, land on a surface, and repeat the process over again. And in fact, it's important to recognize that some of these cells within the colony will produce flagella, which will give them the ability to move on and migrate to a new area. Some of them won't. Some of them don't produce the flagella, and some of them do. And there's actually a difference in the way that the genes are expressed within these cells. So the idea is that some of these cells that are released, often referred to as planktonic cells, sometimes called pioneer cells, they release out, start the whole thing until they form a colony, and then eventually more cells get released and the whole process starts over again. This is an illustration. I call it an animation. It's an illustration. This is a scanning electron microscope image of what this looks like on a surface. This is the very beginning stage of film formation, but that's a real image there, high resolution microscope image. So going back to the figure I showed you a second ago, that would be kind of like right here in the early stages, like you're seeing here. Here we can see a biofilm. This is once again I believe anyway, make sure, yeah. I'm pulling stuff up randomly here, so maybe I'm not sure. Actually, I'm not sure if I'm looking at something correctly there. This is about the same level. This is a biofilm here. This one's a little more progressed than the previous one, if it will focus here. There it is. You can start to see that structure begin to rise up there, kind of like you're seeing here. So anyway, the idea, here's a better one. This is starting to really get thick. Here's the surface, and here's that biofilm really starting to grow, really starting to elevate itself. So the idea is they form this film on a surface. The surface then helps protect them. So let's go back to the PowerPoint here and talk about the benefits here. The benefits come down to the fact that this helps protect them from the elements. You can think of it as like a protective little enclosure. It also helps them stay attached and adhered to one location. And the other thing it's thought to do is help fortify and concentrate nutrients. So the film actually helps kind of recycle nutrients within it, and things like water loss can be prevented and that kind of thing. So it's also thought to enhance communication. Quorum sensing is when microbes actually communicate back and forth chemically, and it's thought to actually make that more effective. And then even plasmid transfer can be enhanced within the biofilm. So that ability to transfer genetic information is done at a higher rate. Other benefits not listed here, as it says at the bottom. But really we're talking about protection, an attachment, and then we're also talking about kind of a concentration of nutrients here. Those are kind of your key benefits. These other benefits are kind of ancillary to those three. Now, when it comes to biofilms in nature, the significance, you can talk about significance in a variety of ways. and significance to one person is not significance to another. So what I like to talk about is not so much what I find interesting about it, but how we can relate this to what you need to know about microbiology. And the way I do that is one of the things you want to know about microbiology, and this is something I haven't really talked about yet. One of the things you want to know about microbiology is that right now they estimate, let me just type this out here. It's currently estimated that less than 1% of microbes can be cultured and studied in the lab. So, of all the microbes that are estimated to be out there, and just to be clear, this is a total guess. No one really knows how many microbes there are, and therefore they can't say for sure how many they can culture. But again, estimated is kind of the key word here. They're estimating that there are millions of microbes out there, and that many of them simply cannot be cultured. And initially, it wasn't known as exactly why, but this has been known for quite some time. That, in other words, we're only studying, or we are not studying. Sorry, I can't type here. Approximately 99% of all microbes. That's a pretty significant number. If you look at microbiology is the study of microbes, and right now we haven't studied 99% of them. Well, what does that tell you? It tells you that microbiology is a very, very early, is a field of study in its early stages, is what that tells you. And it may not look like that because we know a whole lot as microbiologists and scientists. There's a lot that's known, but it's important to recognize that in a relative perspective is virtually nothing compared to what will be known one day. Now, why I talk about that with biofilms is because one of the hypotheses as to why we can't culture these microbes is the fact that they often grow in a biofilm in nature. And a biofilm is this really complex thing, and scientists don't even know exactly what makes every biofilm form. And some of the stuff we were talking about there a second ago is very general. There's lots of specific things that allow this type of biofilm to form, or a different type of biofilm to form, or that allows a particular species to survive within a biofilm. So these are very complex things that are largely not understood. Really just the basics of how it generally works is what's understood. So in order for scientists to get closer to that number of studying closer to 100% of bacteria, then what they're going to have to be able to do is figure out how to replicate biofilms, or how to somehow mimic the benefits of those so that they can get that culture rate higher and higher and higher. And the truth is that number is starting to change because they are already working on ways to do that. And one example of how they're doing that is what's referred to as 3D media. Instead of growing them on a flat petri dish, they're actually growing them in a 3D environment where they're surrounded by media and different substances. And they think that helps give some of these microbes a better natural, realistic enclosure within the environment, or a more natural fit within the environment where they're not just growing on top of something. So in addition, there's lots of other things that they need to do in terms of nutrients, in terms of getting the right kind of balance of nutrients and things like that. But it's important to recognize that really our ability to study microbes in this day and age, right now this is 2016, I'm making it 2017, excuse me, wow, it's 2017. Right now our ability to understand these microbes is really limited, and that's kind of the thing I want to point out, and use biofilms as a potential reason to explain why at least partially that may be. All right, and that's kind of how biofilms protect microbes in nature, and why we're having a hard time getting them to grow in the lab, because we can't quite capture what they're doing there. Now, biofilms are also significant in health and medicine, and the reason for this is that sometimes a biofilm can actually grow inside of a living person, or a living thing. Where this typically takes place is they will form on a non-living material. Biofilms aren't that great at forming on tissue, human tissue, flesh, muscle, bone, things like that. Not as great at forming a film on those surfaces. Where they typically do a better job is on a non-living material. things like catheters, pacemakers, intrauterine devices, hip replacements, all sorts of things like that. They provide a non-biological metal or plastic surface, and these tend to be where the biofilms will form. Once the biofilm forms, then you have the process by which these bacteria are secreting this substance and then trapping themselves within it. And one of the big problems is that eventually the biofilm will rupture, as we show. Let me go back to the picture here. And what will happen is inside your body, this biofilm will rupture, release more bacteria that can potentially go on to cause more biofilms. But even more so, even if they don't go on to form another biofilm, you basically have a source of continual release of bacteria. Here's a figure showing some of the locations where these biofilms will typically form. All right, so inside of a living person, when this biofilm forms, bad things start to happen. The other thing about these is that they are actually quite difficult to treat with antibiotics. Just write that out here. Biofilms do not respond well to antibiotics. So antibiotics are ineffective against biofilm infections in most cases. So the problem when you get a biofilm, not only do you have a continual release of bacteria, not only do you have this kind of ideal growing environment for certain bacteria, but you also have an infection that's not going to respond in a lot of ways to a normal infection in terms of antibiotics. So that all leads to a serious issue. So when it comes to implanting things in the body, this becomes a risk factor that normally wouldn't be a factor otherwise. Now, and as an aside note here, and I honestly don't know much about this, I haven't done some of the research on it, but apparently I've heard a lot of speculation that biofilms may be a part of persistent chronic ear infections. I don't know how true that is. I don't know what other factors go along with that information, but I listed it here as an interesting side note that we know inner ear infections are a real problem for certain kids, and according to some research anyway, they think there might be a connection to biofilm formation. Some of the bacteria that get down in there may form some kind of biofilm, and that may be partly why it's so difficult to treat those and get rid of those. All right.